Good morning and welcome. It is my pleasure to share with you today some of the highlights of the scientific contributions of an American pioneer, a brilliant, strong, soft-spoken, insightful, wise, humble, and highly respected scientist who, despite being a candidate for the double bind of being African American and female, excelled in her career for 40 years. I was fortunate to meet her when I was a graduate student, for her stepdaughter, Rebecca Clark Smith, was one of my best friends, my roommate, and my sorority sister, and she introduced us. I received advice and encouragement from Dr. Daly. I also moderated a national organization for the professional advancement of Black chemists and chemical engineers symposium, honoring her for being the first African American woman to earn and receive a Ph.D. in chemistry in the United States. I sincerely regret that I did not interact with her more often, or let her know how much her achievements meant to me personally. She is a historic person who made significant contributions to her fields and to the community, despite what were probably extreme difficulties. During her career, she collaborated with eminent scientists who were able to recognize her abilities and respect and support her scientific expertise. I will only be able to highlight some of her contributions in the time allotted. Her career timeline is shown. Dr. Marie Daly received 3 degrees in chemistry, a bachelor's in science from Queens College, magna cum laude in 1942, a master's degree in science from New York University in 1943, while still functioning as a fellow and a tutor at Queens College from 1942 to 1944, and a Ph.D. from Columbia University in 1947. Her thesis, a study of the products formed by the action of pancreatic amylase on cornstarch, was based on a suggestion by Professor Mary Caldwell. Dr. Caldwell was also a pioneer who mentored and trained many female graduate students. Dr. Daly's thesis was an analysis of reactivity of the digestive enzymes pancreatic amylase and malt beta amylase with starch and amylose. This thesis project began a long career of investigation of biological materials and cellular interactions using primarily chemical methods. Thus, I would call her also a biochemist as well. A conscientious scholar, Dr. Daly developed experimental techniques where no suitable ones existed. She established the validity of her techniques and then subsequently applied those techniques to address critical and basic issues of the time. After receiving her PhD, she wished to join the prestigious Mirsky Lab at the Rockefeller Institute. Dr. Mirsky could not initially provide support, so she became an instructor in physical science at Howard University from 1947 to 1948, hired by Herman Branson, a noted Black physicist, one of the unsung contributors to the determination of the structure of proteins. His sabbatical collaboration with Robert B. Corey and Nobel laureate Linus Pauling led to calculations which identified the alpha and gamma helical structures of proteins. She then moved to a position as a visiting investigator and a fellow of the American Cancer Society in the Mirsky Lab at Rockefeller Institute in 1948. Dr. Mirsky, who later was inducted into the National Academy of Sciences, and Dr. V.E. Alfrey were 2 highly respected scientists. Dr. Alfrey is considered by some to be one of the forefathers of epigenetics, the study of heritable changes in gene expression or cellular phenotype caused by mechanisms other than changes in the underlying DNA sequence. The Mirsky Lab was then focused on nuclear cytoplasmic interactions, and Dr. Daly published 9 papers as co- or first author with Mirsky and Alfrey. Dr. Daly remained at Rockefeller Institute until 1955, when she accepted a position at Columbia University teaching biochemistry and began to collaborate with Dr. Quentin Deming, a physician, on research into the relationship between hypertension and atherosclerosis. Dr. Deming moved to the recently opened Albert Einstein College of Medicine in 1959, and Dr. Daly joined him at Einstein in 1960 as an assistant professor. She retired from Einstein College of Medicine in 1986. Her contributions include teaching biochemistry and related courses, service in scientific organizations, and establishment of support mechanisms for minority students. He passed away in 2003. The information I will share with you today has been obtained from a careful consideration of her published papers. I believe that highly successful, brilliant scientists would not have collaborated with and published so frequently with Dr. Daly if they were not firmly convinced it was advantageous to them to do so, not because she was the first. Her primary research areas are shown. These two areas will be the focus of my comments today. But first, I'll talk a bit about publish or perish. That's the concept that first appeared in 1938 in an academic publication, and now it's one of the more accepted truisms in academia in the United States. But the assessment of the quality and significance of her research must include consideration of what was known and how science was performed at the time. Dr. Daly published 22 peer-reviewed journal articles in her scientific career, and abstracts of her work were published in the proceedings of at least 12 meetings. 10 of those papers came from her research at Rockefeller on nuclear components and interactions with the cytoplasm, and 9 focused on hypertension, cholesterol, and aging research while she was at Columbia and Einstein. In these days of publish or perish, some people might not be overly impressed with her number of publications, but each of her papers demonstrated a firm and objective grasp of the pertinent literature and a conscientious attention to detail. Her work was distinguished by its quality. She continued to publish throughout her career, despite commitments to teaching after leaving Rockefeller Institute. This next slide is a list of areas of intense investigation, indicating what was not known at the time. In 1944, Avery, McLeod, and McCarty published their classic paper demonstrating that DNA was indeed the cellular hereditary material, one of the most important scientific discoveries of the 20th century. But fundamental supportive and confirmatory work was still being performed to substantiate that finding. When Dr. Daly joined the Mirsky lab, scientists were actively trying to prove or disprove the aspects of the chemistry of the cell which we now take for granted. Dr. Mirsky's lab was heavily involved in this area of research into the interaction and function of nucleic acids, nuclear proteins, and the cytoplasm. The 1953 work of Mirsky, Daly, and Alfrey, which I will mention later, was cited in Watson's Nobel lecture. Her publications were characterized by methodical and conscientious approaches to techniques, frank discussion of experimental limitations, and which did not preclude insightful conclusions that have stood the test of time. Before the demonstration that DNA was the hereditary molecule, the dogma of the time was that proteins were the genetic material, as it was present in the nucleus in nearly equal amounts of DNA and was structurally more diverse. It was easier to imagine a genetic language of 20 symbols than of merely 4 repeating symbols. The tetranucleotide hypothesis proposed that all 4 bases were present in equal molar proportions. The details of the information transfer from DNA to protein were still undiscovered, and many scientists were reluctant to dismiss proteins as being the genetic material. So some critical unanswered questions that she addressed are shown in this slide. The first question is, are there any bases other than adenine, guanine, cytosine, and thymine present in DNA? Is the tetranucleotide hypothesis valid? Do nucleic acids from different sources have the same ratio of purines to pyrimidines? What role do histones play in the chromosome? Does the amount of DNA vary with the activity of a cell? What is the role of RNA? How are proteins synthesized? How does information get transferred from the nucleus to the cytoplasm? All of these questions were still up for debate at the time she joined the Mirsky lab. So her first papers in the Mirsky lab focused on the components, structure, and constancy of the amount of cellular DNA. In 2 papers submitted in 1949, she developed a method to quantitatively resolve 6 purine and pyrimidine components of DNA by starch column chromatography, with recoveries of known mixtures of a 100 plus or minus 4%, and a method that to hydrolyze tissue and qualitatively recover proteins and purines and pyrimidines with minimal destruction. These methods were more quantitative than and accurate than others that were in use. She then asked, are there any bases other than adenine, guanine, thymine, and cytosine present in DNA? And is that tetranucleotide hypothesis still valid? So she took nucleic acids from 11 different organisms as well as from different tissues of the same organism and analyzed their hydrolysates on starch columns. These 2 figures describe some of her results and address the first 3 questions in the previous slide. Please note the clarity of resolution of the different purines and pyrimidines in the figure on the left, and the close correlation of the purine pyrimidine content with the equimolar concentrations proposed by Chargaff's rule in the right figure. These illustrate how the techniques she developed were employed to do extremely precise work. The research concluded that there were no more than traces of other constituents that could be present in those nucleic acids for which a quantitative recovery was made. Shortly afterward, 5-methylcytosine was discovered, and its proportions were found to vary in nucleic acids from different sources, probably explaining the minor variations that were noted in this paper. The tetranucleotide hypothesis proposed that the primary 4 bases comprising DNA were distributed in equimolar proportions in the molecule. This paper showed clearly that that hypothesis was untenable, as was its implicit assumption that all DNAs had the same composition. No great differences in the purine pyrimidine compositions of the nucleic acids was observed, supporting Chargaff's rule that the purine pyrimidine ratio is 1 because of base pairing. The next project Dr. Daly published was in a paper with Mirsky and Rees, and it compared the properties and amino acid compositions of histones and a protamine-like material. At this time, histones were known to be a major component of the chromosome, but little was known about their role in the structural organization of the nucleus. The question addressed was whether the amino acid composition of histones was tissue or host specific. Histone fractions were prepared from calf thymus, calf liver, fowl erythrocytes, and rooster sperm. The histone amino acid compositions were quite similar in each of the samples with one exception. The fowl erythrocytes contained no methionine. It had also been argued that histones were responsible for the continuous structure of the chromosomes. Pepsin was used to determine the relationship of these histones to the structure of chromosomes. Because of the known specificity of pepsin and its inhibition by basic groups, the prevailing wisdom was that pepsin did not cleave basic proteins like histones. This paper firmly refuted that claim and showed that pepsin could digest histones, whether isolated or as part of the chromosome. And in the latter case, the microscopic appearance of the DNA was not altered. It is now established that histone amino acid sequences are some of the most highly conserved in nature. Dr. Daly's next 7 publications represented a shift in experimental methods towards more biological procedures. The first 3, published between 1952 and 1953, focused on protein synthesis in mouse pancreatic exocrine tissue. It had been hypothesized in 1948 that formation of enzyme protein in the pancreas occurred by rapid synthesis of protein, followed by gradual transformation into the characteristic pancreatic enzymes, but there was no experimental evidence before this paper. The mouse pancreas was chosen because of its unique properties. The pancreas can be stimulated to repeatedly cycle through secretion of specific and well-characterized enzymes, synthesis of new proteins to replace the secreted ones, and then a quiescent state until the next stimulus. Rates of synthesis are not complicated by the occurrence of cell multiplication. These cells could undergo large changes in cell function and thus were an excellent model for clarifying the factors involved in protein synthesis. Daly and her collaborators probed the behavior of DNA, RNA, soluble nucleotides, and total protein under these conditions. Despite the large cyclic changes observed in these cells, as you see on the slide, no significant changes in DNA, RNA, soluble nucleotides, or total protein content during or correlating with a complete cycle of secretion and synthesis were found. Next, the metabolism of DNA, histones, and residual protein and their relationship to cellular activity was addressed. Labeled glycine, radioactively labeled glycine, was incorporated into the proteins and also into adenine and guanine, thus labeling both DNA and protein. The rate of incorporation correlated with the metabolic rate of these nuclear components. Uptake and retention of glycine into histones and residual chromosome fractions were compared in the nucleus and cytoplasm of the pancreas, liver, and kidney tissues. In the results of these experiments, uptake is directly proportional to the tissue's metabolic rate. The synthesis of histones is connected to the synthesis of other proteins, but may vary in different parts of the chromosome. Synthesis of protein in the cytoplasm and the chromosomes correlated, indicating that chromosomal proteins were responding to environmental factors. Uptake of the label into cytoplasmic protein was greater than the uptake into residual protein, which was greater than the uptake into histones, which was greater than the uptake into DNA. And interestingly enough, a protein closely associated with RNA was found in the pellet that was obtained and prepared from the pancreas. This pellet incorporated labeled glycine at about 3 times the rate of the total pellet protein. Observations about RNA were an early clue to the role of cellular RNA in protein synthesis. In the results of this paper, Synthesis of Protein in the Pancreas, which was published in 1953 and is her most often cited paper, she addressed the role of this ribonuclear protein in the cell's microsome fraction in cellular protein synthesis. And this is the paper that was cited in Watson's 1962 Nobel lecture because of its observation of the importance of RNA to cellular protein synthesis. Mice were fed or fasted, injected with glycine with the 15th isotope of nitrogen, sacrificed at various times. Organs were removed and homogenized and centrifuged at low speed. The supernates were then centrifuged in an early use of high-speed centrifugation to pellet the microsomal fraction containing ribonuclear proteins. The pellet was analyzed for multiple factors, radioactive glycine, phosphorus, RNA, amino acid composition, and zone electrophoresis of RNA was done. And they found that ribonucleic acid and protein form a stable complex in the microsomal pellet. The amount of RNA correlated with the rate of protein synthesis, with the rate of uptake of label by protein, and with the level of uptake of label by total protein. The time course of uptake indicates that the pellet protein is the precursor for the synthesis of these secretory proteins. Nuclear protein, therefore, they concluded, and the protein from the pancreatic microsomal pellet are part of the protein synthetic machinery. And I remind you that one of the questions was, what's going on when the cell does protein synthesis? The uptake of C14 alanine by these preparations is impaired by pre-incubation of the microsomes with ribonuclease, direct evidence for the requirement of RNA for protein synthesis in the microsomes. Added purified pancreatic enzyme precursors do not serve as precursors for the proteins synthesized by the microsomal fraction. And so that last conclusion, as I said, is direct evidence for the dependence of protein synthesis on the presence or intactness of RNA, a very significant finding. It was published 2 years before Daly reported his observation of ribosomes, for which he won the Nobel Prize. Siekiewicz had shown that protein synthesis occurred, but this paper showed that using Siekiewicz's system, that destruction of RNA strongly inhibited protein synthesis. Particles had been seen earlier in the microsomes by Smelly et al., but these experiments indicated that the particles were a site of protein synthesis. So these authors proposed that the microsome is a combination of protein synthetic machinery and that is involved in synthesis of the tissue proteins. In a recent review article summarized in this slide on the role of dietary and blood cholesterol in atherosclerosis, this review article describes the evolution of understanding the effects of dietary exogenous cholesterol on atherosclerosis, and it supports the lack of correlation of dietary cholesterol with atherosclerosis. It was published in 22, it's a review, and that's an observation that Dr. Daly made in 1957. Atherosclerosis is the buildup of fats, cholesterol, and other substances in the arterial wall, but it's a controversial area since dietary guidelines of 300 milligrams per day of cholesterol was set arbitrarily in the 1960s. And when further studies were done in the 1990s, there was no correlation seen between dietary and blood cholesterol in studies. So multiple large cohort studies then were done since the 1990s, and they have led to the elimination of dietary guidelines on the upper limits of cholesterol in 2015. And as I mentioned, this was addressed in a series of papers by Dr. Daly and Dr. Deming in 1957. She began research into factors contributing to heart attacks when she accepted the position at Columbia University. Her first publication in this area, as I mentioned, occurred in 1957. Her research progressed from a study of the influence of hypertension on dietary-induced atherosclerosis in rats to detailed studies of the synthesis of lipids in rat aorta, and it resulted in 9 papers. In 5, she was the first or only author. Atherosclerosis could be induced in rats by dietary cholesterol supplementation. And hypertension was known to increase the severity of atherosclerosis in both man and experimental animals. Most observers attributed this observation to local damage caused by elevated blood pressure or turbulence. A critical question was that she sought to address whether the changes seen were a local effect of elevated blood pressure or increased lipid deposition. Abnormalities in serum lipids had also been observed with atherosclerosis. So Dr. Daly began by asking if hypertension modified the rate of development of atherosclerosis, dietary-induced atherosclerosis. The effects of hypertension on the serum concentration of cholesterol and the synthesis of lipids in the aorta were investigated in vivo and in vitro. If you look at the slide, the hypertension was induced chemically and surgically in mice, and after a month, mice were placed on a cholesterol supplemented diet. They looked at blood pressure, blood and tissue, cholesterol levels, rates of cholesterol synthesis, total serum lipids, and formation of lesions. And they looked at that over periods of 6 to 20 weeks. Normotensive rats on the diet did develop increased serum concentrations of cholesterol, total lipids, and beta lipoprotein, and increased content of cholesterol in the tissues, as well as atherosclerotic changes. But if the rats also had induced hypertension, that intensified the effects of the diet. in producing lesions, hypercholesterolemia, hyperlipemia, and increased cholesterol in the liver and carcass, despite the fact that endogenous cholesterol production ceased on the diet in both hypertensive and normal rats. This reveals a potentiating effect of hypertension on atherosclerosis. In typical Daly fashion, she investigated multiple potentially pertinent parameters: respiration, cytochrome oxidase activity, cholesterol synthesis and concentration, lipid composition, glucose utilization in these induced hypertensive rats, and arterial lysozymes and connective tissue in atherosclerosis and hypertension in rhesus monkeys. Realizing that the investigation of the aorta should be performed on the intimal and the medial portions of the vessel, as the outer adventitial area was primarily structural in function and atherosclerotic lesions are primarily intimal, she developed a new method for obtaining medial and intimal tissue free of adventitia for in vitro studies. Her studies in this area of atherosclerosis and hypertension indicated that dietary supplementation or ingestion of cholesterol were not the most critical factors, that cholesterol was actually made in the arterial tissue and was a significant factor contributing to atherosclerosis. In summary, Marie Daly's legacy is not only that she was the first Black woman to receive a Ph.D. in chemistry in the United States, but that she was an excellent scientist who made significant contributions to the field, despite the obstacle of the double bind she must have experienced. Despite her unique situation, we all know that her ability to do so required the critical support of some of the most respected scientists of her day. Those types and levels of support are never easy to obtain or validate. They validate what a special human being she was, worthy of the many honors that she received in her professional life and continues to receive on the occasion of this landmark event. I thank you for your attention and interest today. Dr. Daly's scientific life was and is an inspiration and a source of pride to all, but especially to those of us who are minority and female and victims of the double bind. Despite enormous odds, she persevered, excelled, and retained her dignity and integrity, and I salute her.